Good morning and salam sejahtera. My name is Chee. I'm going to present my proposal. The title of my proposal is Diatoms Diversity Associated with the Decomposition of a Partially Submerged Buried Cadaver in Stagnant Water Body. I was supervised by Dr. Siti Safar Ismail and co-supervised by Dr. Nurul Shahida Rizwan. Nowadays, crime rate increased drastically, especially homicide cases. The accurate postmortem interval determination and find out the location of clandestine grave can be the important evidence to arrest the criminal. Therefore, we decide to study the ability of diatoms to be used as forensic tool to collect evidence from crime scene, as diatoms diversity is highly abundant in atmosphere. There are three problem statements for this study: difficulty of the determination of postmortem interval and find the location of clandestine grave, lack of advanced technique to collect evidence from soil, and lack of information for clandestine grave buried in partially submerged condition. The two objectives of this research are to determine the diatoms diversity associated with the partially submerged buried cadaver in a stagnant water body and to elucidate the fact of a partially submerged buried cadaver in a stagnant water body. Significant study of this research are to locate the clandestine grave, discuss about the diatoms with adhesive characteristic to use as forensic tool, and collect useful evidence at crime scene. Temperature, moisture, soil pH, and availability of oxygen are the factors which can affect the decomposition rate of cadaver. The slide shows the complete decomposition stage, which mainly as initial decay, putrefaction, black putrefaction, woody fermentation, and dry decay. To conduct the burial experiment, 20 grams of fatty flesh is prepared and placed into a clean terracotta pot and filled with 2 cm depth of soil. The mass of the fatty flesh will be recorded. The pot is then partially submerged, buried in a soil environment of stagnant water body with a depth of 15 cm. The fatty flesh will be buried into several designated sampling pots. A control burial pot will also be prepared. The slide shows the procedure to extract diatoms and prepare diatoms slide. About 10 grams of topsoil will be collected and placed in a centrifuge tube, and followed by 1 ml of 1% formalin. The mixture will be spun at 2300 for 15 minutes to remove excess formalin. Then distilled water will be added to depth of 0.5 cm of each diatom sample in centrifuge tubes for agitation. Half of the agitated sample will be removed in a plastic bottle which contains formalin and stored as fresh sample. Potassium permanganate solution will be added to the remaining agitated solution and left for 24 hours. After 24 hours, 1 ml of concentrated hydrochloric acid is added into the solution and placed in a water bath with temperature of 80 degrees Celsius for 1 hour. The sample will be centrifuged at 2300 rpm for 15 minutes after taken out from the water bath. The supernatant will be siphoned off and distilled water will be added. This centrifuge step will be repeated for 6 times to get a clean diatoms sample. To prepare diatoms slide, approximately 1.5 to 2 ml of diatoms sample will be placed on a cover slip of the slide. Then distilled water will be added to the sample on the cover slide to spread the cells. After 15 minutes, a drop of potassium iodide will be added to the diatom sample and the sample will be left for 2 hours. The slide can then be observed. The atoms will be analyzed under magnifying microscope, scanning electron microscope (SEM), and transmission electron microscope (TEM. The expected result of this research are the decomposition rate, diatoms diversity, and soil pH will increase in early decomposition stage and decrease in late decomposition stage. The reason of decomposition rate decreases is because the soft body tissue is completely decomposed in early decomposition stage and left bone only. This causes the diatoms diversity to decreases as the surface area of flesh decreases and results the disarticulation of diatoms. The soil pH decreases because the ammonium ion released by the cadaver will be used for nitrification and produce nitrate ion and thus alkalize the soil. That's all from me. Thank you.